So it's a pleasure to be presenting virtually to the conference this year. I'm going to talk about some of the work that we've been doing on the role of pharmacology in selection of COVID-19 candidates. Fair to say there are many more questions than answers at the moment, so that's very much going to be the theme of my presentation. So I'll start with just some of my disclosures. So it's clear that the lungs are a primary target organ for infection, for replication, and for the disease phenotype. But we do know that ACE2 is expressed in many other organs, including the kidneys, the gastrointestinal tract, the central nervous system. SARS-CoV-2 RNA can be detected in stool specimens of patients for protracted periods of time, in some cases even after the virus has been cleared from the lung. The virus induces acute tubular damage in some patients. We also know about some patients exhibiting liver comorbidities. So I guess the first question that we were addressing is, are therapeutic drug concentrations within the systemic circulation and other affected organs going to be required for successful therapy and PEP? When we start to think about chemoprevention, drugs may need to penetrate into other compartments, and they'll need to do this at sufficient concentrations to inhibit viral replication. And we started thinking through some of the issues around some novel PK compartments, such as the mucous membranes present in the nasal cavity, in the oral cavity, the throat, the surface of the eye, tears, and the upper respiratory tract and lungs. Another question of, well, reducing the inoculum size, even if we don't completely block transmission, provide any benefits for morbidity and mortality. And there's some evidence that higher SARS-CoV-2 RNA is indicative of a poor prognosis, and some very weak actually evidence that for other pulmonary viruses, that the size of the inoculum to which the patient is initially exposed may be associated with disease severity. Now, there's been a number of calls for the application of clinical pharmacology principles to SARS-CoV-2 candidate selection, and some of these are summarized here from the British Pharmacological Society as well as other major groups. And many of these have focused on candidate selection, others on the safety of the putative repurposing opportunities, and then other work emerging on trying to understand the drug interactions for COVID-19. One of the things that we've been doing recently as part of an international group across the US and Europe is trying to better articulate some of the consensus that has been achieved for protein binding in other antiviral programs which we've all worked on, such as HIV and HCV. I think it's fair to say that there's been an under-recognition of protein binding in the early trials that were conducted for SARS-CoV-2. But I think we're in the danger now of moving into an area where there's over-interpretation of protein binding. I think it's very important to underscore, as we all know from our work with other viruses, that the impact of protein binding requires an empirical determination and cannot just be assumed from the free drug fraction in patients. If we think about maybe some of the other principles of antiviral pharmacology that we can apply from other viruses, well, we know that successful regimens have utilized drug combinations for HIV and for HCV, and that we usually target one or more mechanisms within the viral life cycle for maximal antiviral effect. In other viruses, successful regimens have been developed on the basis that their plasma or target site concentrations are fully suppressive across the entire dosing interval. So we're more accustomed to thinking about C trough as a metric rather than C max or AUC. But it's important to recognize that the absence of empirically determined protein binding adjusted EC90 or EC95 values complicates our current interpretation of the existing anti-SARS-CoV-2 activity measurements. So one of the first things that we did back in March and April was we looked at all of the antiviral candidates that were out there, and we tried to benchmark them on the basis of their plasma exposures in comparison to their in vitro activities. Now, at the time, there were 72 entries in the database with 57 derived EC90s. Unfortunately, most people are reporting EC50 and not EC90 or EC95, and derived EC90s where we could. And since for many of the drugs being investigated, they're not HIV drugs, and the main pharmacokinetic parameters that have been reported are Cmax or AUC. So we did an initial investigation of the Cmax to EC90 ratio where available. This database is currently being updated. We now have 430 entries with 134 derived EC90s. But we've taken a tiered approach to candidate selection where we looked at this crude analysis of the Cmax EC90 ratio, then moved into an assessment where available of the full PK profile relative to the in vitro activity, and where molecules were close to achieving those predefined concentrations. We went back and applied modeling approaches to see if we could simulate optimized doses or optimized schedules, which would maintain our trough values above the EC90 targets. There's lots of caveats in this type of approach, so please do read the limitations carefully in our published paper. Now, one of the things which was obvious from this analysis is if you rank candidates purely on the basis of their in vitro activity, you get this list of most potent molecules, including drugs we're familiar with, such as Remdesivir, hydroxychloroquine, some of the other 4-aminoquinolines. But this actually tells us nothing about the pharmacokinetics from the approved dose of the drug. And of course, most of the repurposing opportunities which are being explored are directly repurposing the approved doses for SARS-CoV-2. Actually, when you look at the ranking in terms of the Cmax to the EC90 ratio, you get a very different list of drugs which appear to be at that point where the plasma concentrations for at least some of the profile exceed those concentrations which are needed for activity against the virus in vitro. And just a couple on this list that I want to flag up because I'm going to talk about them in more detail is the Remdesivir, the Nitazoxanide, as well as Declatazvir. So nitazoxanide was a molecule which we looked at very early on back in April. There's quite a lot of preclinical in vitro data supporting this molecule now. The initial report was by Wang back in 2020, but it's been confirmed by several groups, including for the major circulating metabolite of this drug. Activity has also been reported by the manufacturer of the branded product in the US against other viruses such as bovine, canine, and human enteric coronaviruses, as well as influenza. The mechanism of action for SARS-CoV-2 has not been investigated, but for influenza, the drug appears to interfere with the N-glycosylation of the hemagglutinin of influenza. And it is interesting to note that SARS-CoV-2 spike protein is also heavily N-glycosylated, is also a heavily N-glycosylated protein. There are several putative secondary mechanisms for action for this drug. It's been reported to amplify the host innate antiviral immune response for other viruses. And it's an antagonist of this ion channel in the lung, which may result in bronchodilation within some patients. There's actually some clinical supporting data for nitazoxanide as well. Single doses of this drug, up to 4 grams, have been studied in humans without major safety issues. It has excellent safety at its approved dose. It ranked very highly in our analysis of Cmax versus activity. Quite interestingly, back in 2014, a 600 milligram twice daily dose of this drug reduced the duration of symptoms and virus burden in a placebo-controlled phase 2b/3 trial of uncomplicated influenza. Now, these weren't profound impacts on influenza, but it did provide a signal that there may be for this a benefit for this drug for another pulmonary virus. There's also some data conducted over a decade ago for hepatitis C virus. Importantly, though, we've done a lot of simulations with this drug, looking at the active metabolite, and those simulations indicate that the doses may not be optimal for influenza or HCV. So I think there may be scope for improving application of this drug through dose optimization. So just to illustrate that point, this simulation here shows the pharmacokinetics of tizoxanide after administration of a 600 milligram BID dose of nitazoxanide. And you can see the target concentration here set as the EC90 against influenza, and the drug barely creeps over at Cmax those concentrations required for in vitro activity against the strains of the influenza virus. We did some dose simulations for this drug against the reported anti-SARS-CoV-2 EC90 values, and these predictions indicated that doses closer to 1,500 milligrams or 1,400 milligrams twice daily or 900 milligrams 3 times daily would provide concentrations in plasma above the in vitro EC90 in 90% of the simulated population across the entire dosing interval. And we're currently in the process of trying to move this compound into a phase 1, seamless phase 1, phase 2A trial in Liverpool and South Africa, looking at these higher doses in the Agile platform trial. Now, there's been some recent interest in sofosbuvir and declatazvir for SARS-CoV-2, and this interest has come predominantly from several small trials which were conducted in Iran. This is interesting research, but we have to be aware that many limitations of these trials, which include small numbers, issues around randomization for some of the work, no placebo, concomitant standard of care with bevirin or boosted lopinavir, and no viral endpoint included in the trials. But there is a signal of a benefit from a meta-analysis. Now, the in vitro data for declatazvir currently come from a single source preprint. Now, it's important to note that there are multiple reports for sofosbuvir in the literature, but the other reports demonstrate lower or no activity of this drug against SARS-CoV-2. Just to summarize the in vitro data, the declatazvir appears to have activity similar to that of chloroquine, and the sofosbuvir appears to have activities in the region of lopinavir and ritonavir. But this group did demonstrate activity across a range of cell types, including the viral cells as well as human models, which are thought to be more relevant. Now, we've looked at the pharmacokinetics of sofosbuvir, and this analysis does indicate that at the approved dose of 400 milligrams, neither the sofosbuvir itself nor the major circulating metabolite, the dephosphorylated active form of the sofosbuvir, reach concentrations in the plasma which are anywhere near these concentrations which have shown to be active in vitro. So there may be some other mechanism at play there for this drug, but it doesn't seem very likely that this drug is going to reach target exposures required for viral suppression for SARS-CoV-2. We did look at Declatasvir in much more detail than for sofosbuvir. Declatasvir simulations indicated that target concentrations, trough target concentrations above those concentrations needed to suppress the virus in vitro may be achievable. But this would require markedly higher doses than the approved dose. And our simulations indicate to keep the population above this C-trough target, you'd require doses closer to 330 milligrams 3 times a day in order to hit this target. Of course, we don't understand the exposure-response relationship for SARS-CoV-2, and I'm sure we've got a lot to learn about this over the coming months and years. I did want to just say a little bit about Remdesivir. I think it's interesting to compare plasma exposure response across the ProTide antiviral nucleosides. So I've just compared here the indication for TAF in HIV, sofosbuvir in HCV, and then Remdesivir and sofosbuvir against SARS-CoV-2. Now, this class of drug is beneficial because it has a high penetration into the target cell, but we have to be mindful that the EC50 or the EC90 is an extracellular metric. It's not an intracellular metric. So it is correct to compare those EC50, EC90 values to the plasma concentration or the tissue concentration and not to the intracellular concentration, because the intracellular active form is different to the value presented for the EC50 or EC90 value. Now we know that in HIV for TAF, the prodrug exceeds those EC50 values by orders of magnitude and is maintained for a short period. But of course, this high intracellular concentrations and this long intracellular half-life then maintain the activity across the dosing interval beyond that. Similar for HCV with sofosbuvir. You can see with Remdesivir, you do again exceed the in vitro target concentrations early on, not to the same degree as you do with HIV and HCV, but again, this demonstrates the point. And when we make that comparison with sofosbuvir again, the target concentrations do not exceed this extracellular metric of the EC50. So some other thoughts about antiviral development that we've been looking at. This is a paper we've submitted recently, chasing COVID-19 chemotherapeutics without putting the cart before the horse. I think it's important to recognize that a global pandemic requires a global solution, and equitable access to the emergent medicines are going to be critical. Now, this requires thought during development about compatibility of the products with the global healthcare systems, which is also influenced, of course, by the use case, whether it's prevention, whether it's post-exposure prophylaxis, or whether it's direct treatment. And of course, when in treatment you deploy it, whether it's early in disease or whether it's in late stage disease. Relying, I think, solely upon pre-existing formulations and posologies optimized for other diseases carries inherent risk. We accept that advanced formulation development may take longer to implement than a direct repurposing, but I think it seems likely that this may improve our success rate in finding repurposing opportunities for COVID-19. It wouldn't be me if I didn't mention long-acting drug delivery, and we have been looking at opportunities. Clearly, you need effective drugs before you can think about compatibility with long-acting, but I think it is important to note the potential benefit of a long-acting drug delivery strategy as a bridge towards a vaccine. And back in May this year, results from the HPTN-083 trial in HIV chemoprevention clearly demonstrated that long-acting cabotegravir was much more effective at prevention than an oral prevention strategy. So I'll leave you with more questions than answers, as I promised I would do at the start of the talk. And they're summarized here. Are therapeutic drug concentrations within the systemic circulation and other organs a prerequisite for successful therapy? We're reducing inoculum size without, even in the absence of completely blocking transmission, provide benefits for morbidity and mortality. Do we have time to wait for steady state pharmacokinetics to achieve? So for HIV, we accepted in some cases 1 to 2 weeks to reach steady state, but in SARS-CoV-2, that's really going to be too late, because the antiviral really need to be starting to work on day 1 of therapy. Are drug combinations going to be a prerequisite for success? Should we be thinking now about potential future issues with drug resistance as we consider this emergency development phase for therapeutics that we're going through? Are the current methods for viral quantification fit for purpose, and can we even use them to define minimum effective concentrations in patients once we do have more successful drugs coming through the development plans? So I leave you with some acknowledgements. I do want to acknowledge my team in Liverpool that have been working almost 24 hours a day for the last 6 months. Joe Sharp, Megan Neary, and Helen Box, who have been working around the clock to establish SARS-CoV-2 animal infection models, which we're starting to do our first work in now. Paul, Curley, Lee, Titham, Usman, Arshad, and Anthony for working around the clock on in vitro DMPK for some of these targets, as well as optimizing innumerable bioanalytical assays to underpin our preclinical portfolio. Rajit Rajoli and Henry Pertners, who have done an enormous amount of pharmacometrics work over the last 6 months.